I'm just going to talk about a few thoughts about dose control, and mostly in the context of TDCS, because that's what we've been looking at. But I think most of these points I'm going to raise, they're going to apply to pretty much any form of stimulation. Of course, good to remind ourselves why we're here, and I think in part this is because we are all aware that the effects of TDCS, but lots of the other techniques we use are highly variable. These are just a few of the examples going back quite a few years now. Rovereto showed this for different types of TDCS, and then people from other groups like Pablo CIMeC and so on and so forth showed that there is often not a really good consistency among the results. And as mentioned by Hartwig already, Jonker and colleagues have done a very impressive study, again showing that the effects of TDCS are highly variable. And so the question is what we may be able to do. I'm sure there are many things one can do to address this, but I'm going to focus simply on dose control. And there are probably different ways to do dose control as well, but I'm mostly going to focus on using current flow simulations to get an estimate of where we may be delivering current to, how much we're delivering of it, and so on and so forth. And therefore to give us a prediction of what we're actually delivering. And the point here is that it seems a good starting point. Hartwig mentioned that there are a lot of variables that influence what happens, but ensuring that we deliver what we want to deliver in the first place and knowing where we deliver to seems to be quite relevant. So that's the main approach we're going to explore here. Now, the point is that these are simulations. They're not necessarily telling us about the ground truth there, but they're probably the best approach we have, at least non-invasively at the moment. Now, as many of you will be aware, when we apply TDCS in a given montage, then what is actually being delivered to the brain is likely to be very variable. And that's both in terms of the distribution of the current, but also in terms of the intensity. Now, this is a study from a few years ago by Laxo and colleagues. What you see here are different brain scans on which current flow modeling was conducted. And if you can see that, if you look very closely, both in terms of the target location, which was M1, but also in terms of where the peak is, the intensity is really quite profoundly variable. And it's probably in the order of 100% of difference between the lowest and the highest values that you get here. So that seems to be quite dramatic and quite consequential a priori, that if we apply the same intensity, I think in this simulation it was 1 milliamp, we may actually get a variance of up to 100% in terms of what is reaching the cortex, and particularly the area we're looking at. This is a more recent study which kind of looks at that again, comparing different montages and really asking sort of how is the distribution of current with these, but also what's the variance here, and this is expressed in the lower panels in terms of the standard deviation. And again, you can see that it can really be quite profound. Now, we all would accept that it's the current that we deliver that causes physiological changes, and that's ultimately what then causes the behavior that we're interested in. So dealing with such a huge source of variance at the very start already, I would argue is already a big contributor to the problem that the behavioral outcomes we see can be highly variable. Now, It's been argued many times that we can use current flow modeling to optimize stimulation and to control the dose of what we're delivering in an individual and across individuals. But it's actually not entirely clear what we should optimize for. Again, Hartwig has nicely made the point here. It's appealing to think, well, if we just make sure that someone gets, across subjects, we get the same intensity to a specific area, seems to be a good starting point, but we in fact don't know if that helps us to optimize stimulation and to increase effect sizes and to reduce the variability across subjects and studies that we often see. So we don't really know whether that's the best way, but even if we control intensity, then inevitably we still haven't controlled for the focality of the stimulation. And by that, because TDCS isn't focal in any case, I just mean whether we constrain the extent of stimulation more or less. And also we haven't controlled for the direction of current in a particular area or even a range of areas. And as I think we've heard this morning, that is of huge consequence as well in terms of what actually happens to the underlying neurons and the physiology and therefore the behavioral effect. So it's by no means clear what should we actually do. And I'm just going to give a few examples of these three possible points that one should optimize for if one wanted to control the dose. Suffice to say, I don't really have final answers, but I think these are all points that are worth discussing in a workshop like this. So when we think about controlling dose or individualizing dose, I think at a very simplistic level, what it really means is that we don't want to just fix the knob on the stimulator that says deliver 1 milliamp or 2 milliamps or whatever to all subjects, because as I just said, we know that what actually then arrives in the brain is hugely variable. What we want to do is we want to control the parameters of stimulation with the idea that everybody gets the same in terms of what's delivered. And again, that could be the intensity, the extent, or the direction. So we have a good idea that in terms of intensity, that may be a useful thing. This study that goes back quite a long time by Dylan Edwards and Maren Bixen. This is using transcranial electrical stimulation, so 2 electrodes put over motor cortex and then high voltage pulse to elicit an MEP. This is really quite unpleasant, which probably explains why these authors only used 3 subjects. But it actually is also highly reliable in terms of the evoked MEP, because the electrodes are basically attached, glued to the scalp, and they give highly consistent motor evoked potentials. Now, when you adjust the intensity to a fixed output, which is a bit like what we do with TDCS, then on the right-hand side here, you can see the evoked response in those 3 subjects and what the likely field distribution is hugely variable. One subject doesn't actually have an MEP, the others have fairly large but still quite variable ones. Now, when you then adjust the stimulator output so that each subject gets the same MEP, you can see that that is really quite effective. So you can tweak the stimulator to deliver the same, to cause the same effect. But what that means is each of those subjects gets a very different stimulus. So the top subject, the poor subject at the top, gets nearly twice as much in terms of stimulus intensity than the lower 2 subjects. But it just gives an indication that with these kind of approaches, we may be able to control what's being delivered, and that may actually have an effect on the outcome of the measure that we're interested in. Now, this was TS. With TDCS, we have much larger electrodes, and they're generally much further apart, so we're likely to stimulate a big area. But as a starting point, we could just look at what's actually being delivered to the target area. So in this case, it's shown here, it's the motor cortex. And we could just say, well, with the standard approach of, say, 1 milliamp or 2 milliamp, as shown in blue and green here, what is actually arriving in the motor cortex in terms of the intensity? And as you can see here from these distributions, and as already implied by the Laxo paper, what actually arrives is hugely variable. So it's in quite a profound spread of, again, close to 100% in terms of the intensity that arrives in the target area, ignoring what happens everywhere else. But if we now adjust the stimulus output so that each subject gets a target intensity, then we can remove that variance altogether. What that, of course, means is that we now have variance in the stimulator output. But that's just what we determine, and that's something that we can live with. So in a way, if the target area was really the only determining factor, we could control quite well in terms of the intensity what we deliver there. But of course, that's not the only place where current is delivered to. We could look at the distribution. Again, as you see at the top, with the 1 milliamp fixed dose or fixed intensity, we get a fairly variable distribution of current. You can see this is a probability map of 50 subjects. We see that in the target area, we don't actually have a consistent intensity, and that there's quite a lot of variability. Now, if we fix the stimulator output so that every subject gets the same intensity at the target area, not surprisingly, that variance at this point is removed. But as you can see, that doesn't remove the variability in other areas. And in fact, as you can see, you may even get consistent but more distributed current delivery in nearby areas as well. And to anticipate anything that I will talk about subsequently, how we can control for this distribution isn't really quite clear yet, I think. Now, one important point is that because with a fixed intensity of stimulation, or fixed stimulator output, I should say, it's so variable what is delivered, we can easily create the situation where in a given study, which uses, say, 1 milliamp, the population is actually receiving a relatively low intensity, and in another study, the population may receive quite a lot higher stimulation, just because of the sampling of the population, and this becomes particularly important with small sample sizes. So this inherent variability can easily create the case where two studies that give the same output in terms of what's set at the stimulator effectively stimulate at quite different intensities on average. And again, it's not difficult to imagine that that could be quite a profound source of variance in terms of the physiological effects and the subsequent behavioral effects. In a way, this speaks for having larger sample sizes when we can't control what we deliver. But equally, the point, of course, is that ideally we would like to be able to control what we're inputting into the brain. Now, what about focality? So with TDCS, that's a bit of a concern. There are some techniques to constrain the stimulation spatially. I think somewhat counterintuitively, it's called high definition. It's certainly not spatially precise, but it constrains the delivery of current better than bipolar montages. So perhaps if we increase the focality, we can decrease variability as well. And we can look at that with current flow modeling again. And the point is, not surprisingly, with HD TDCS, we get a much more constrained distribution of current, which may be helpful. We don't really know, but we often think that being more focal is actually a good thing. But when we do this, actually, we still see that at the target area in M1, there's an enormous variance in terms of what actually arrives at that location. And not just that, with the same overall stimulation intensity, we now actually have equal variance in terms of what's being delivered, but also a lot weaker stimulation. So you can see the mean of these two distributions. This is for bipolar. The purple is for HD. Given the same stimulator output, we now actually deliver weaker current. So it's spatially more constrained, but it's going to be weaker, at least at the target area. And if we wanted now to counter for that, then most likely the extent of stimulation above a certain threshold would grow again. So we'll be losing some of that advantage now in terms of spatial specificity. And it already points to something quite annoying, and that is that almost whatever we do to control, it has some cost function and some perhaps undesired side effect. So in this case, if we constrain stimulation, it becomes weaker. If we counter for that, it becomes less spatially precise. Of course, one question is, does that make any difference? And this is something I won't show too much data about, but that's really the overarching, or the main pot of gold that we are all going for. Now, if we do whatever we do to control stimulation, does it have any effect, or could we just not bother? Now, in this case, if we compare those two types of stimulation, it doesn't actually look like there's a huge benefit. So if we apply a fixed intensity with HD TDCS, we see an effect in terms of the subsequent MEP change. And if we do it individualized, we actually seem to get a fairly similar effect in terms of the mean and the distribution. Now, this is a single study or data set, which we are doing some replication on, but it certainly means that that idea that we can simply just control the intensity at the target side and therefore reduce variability or increase effect sizes is at least not straightforward. So the last bit is to talk about direction. As we heard this morning, it's quite important with this type of stimulation what the direction of current flow is with regard to the underlying neural elements. And so perhaps we should focus more on that. The actual intensity may or may not be relevant. And the question is direction a relevant factor that we ought to control or that we could control? And the point has been made this morning that depending on how current flows with regard to the orientation of a neuron, but that has a huge impact on the physiological consequence, whether there is net soma polarization or hyperpolarization, a depolarization or hyperpolarization, or whether anything is happening in the first place. Now, this is, of course, a nice thing to think about in a model neuron like here, or in an in vitro setting. But then if we move to the human brain and the type of stimulation we apply through scalp electrodes, it becomes a lot more complicated because unfortunately our brain is contaminated by the gyral structure. That's for the things we want to do here, hugely annoying. What that means is that neural elements in different parts of a sulcus and gyrus will be actually, or most likely, affected by the stimulation in very different ways. Neurons that have are exposed predominantly to tangential field will perhaps have a lot weaker physiological change, or none at all. And neurons that are exposed to radial field have a much stronger effect. But then the problem is that in two adjacent walls of a sulcus, if you just think about the orientation of the pyramidal neurons, they would basically experience the opposite polarization with a given type of stimulation. So if current flows this way, then you can see the effect here. And say if this was the primary motor cortex and the primary sensory cortex, then it becomes quite interesting to think about what that means, because neurons in the motor cortex would be polarized predominantly in a specific way, and neurons in primary sensory cortex in the opposite way. We know these areas are very intimately connected. So in terms of the net effect we measure with MEP, EEG, and whatnot, it probably becomes very important to think about this, how those polarization effects conspire to the net effect we see. And that's something I think is quite important to think about in M1S1, but really in any part of the brain. So the idea that we have a uniform effect of stimulation is, I think, quite simplistic and most likely not true, but really raise the point if you look at this. So what are we going to control for now? Can we control for this orientation? The problem in terms of variability, of course, is that in each subject, the montages are slightly, the electrodes are slightly differently positioned with regard to the specific orientation and position of the target area, let's say the primary motor cortex or central sulcus. And therefore, the neural elements we want to target are likely to experience slightly different current in terms of its orientation. And again, that could be a very likely source for a lot of the variability we see. So some indication that controlling the direction may have an effect or may make a difference, whether good or not, and this is work from Ravji and colleagues. Here they compared bipolar montage where you'd try to send current across the central sulcus and M1, and this is the example here, and they called it posterior anterior TDCS. And then an orthogonal montage where the current is directed along the sulcus. And so if you, again, consult current flow modeling, you can see that that seems to have quite makes quite a big difference in terms of the direction of current with regards to the central sulcus. Whereas in the posterior anterior case, you seem to get a fairly consistent effect in the wall of the central sulcus. It's really not the case in the mediolateral orientation. But you can immediately see already here as well that this is, of course, sort of approximation. There are no right angles in our brain, so you know, you can see even here this is hugely dependent on the curvature of that individual. But when Ravji sort of tested this and said, well, does it have any appreciable impact on the effect of stimulation? The answer seemed to be, yeah, it does make some difference. So when you use this posterior anterior montage, you seem to get an stimulation effect in terms of the modulation of MEP size. In their case, it was a reduction. In the mediolateral case, that didn't seem to be the case in a very consistent manner. And there was a hint for the effect being slightly less variable as well, which is another point that, of course, is important. So controlling in a simple way for the direction of current flow may be quite relevant. So this could be something to control for in terms of, you know, still considering as a way of dose control or what is actually being delivered. Now again, it's illustrated here again that the point I think that is quite important, I think, in terms of what the net effects are, is that when you look at motor cortex and the opposite sensory cortex, the polarization effects may be exactly opposite. So in the sense that when you think about the angle of the current entering the cortex, it will be basically almost the opposite in M1 was S1. This is just two different ways to express that, as shown here. And here you see sort of the histogram of that in terms of the orientation of current and with regards to the surface of the cortical target area. And so the idea that we can control the direction of current flow is appealing, but also there's a lot of complication to think about. But another point from this, of course, is that with almost all the approaches we use at the moment, this will always happen. So when we have an area that we target, we must be aware that most likely the polarization effects are the opposite in very nearby areas that just happen to be on the other side of that sulcus. Now the question is, could we control for that in the sense that we get the same polarization effect in both areas, both sides of M1 and of the central sulcus? Again, we can try that with current flow modeling. So the idea would be with conventional TDCS, we probably get opposite polarization effects, but can we somehow create a case where the direction of current in both sides is identical, which then would be quite interesting to test for what the functional consequences of that are. And we don't know, but it is something that I think people hasn't really been looked at. Though the short answer is yes, we can do that. We can optimize our montage such that current in our target areas has predominantly the same direction. Now, the way that one can do that is by basically saying, find the best solution for this target that I specify, that current flow should be more or less having the same orientation. Now, I talked briefly about costs of any way we optimize things, and the cost here is that means in each individual, we now have a very different montage, and we can only really do that if we use multi-electrode arrays, and that is something that isn't very practical. But you can see here, for that specific optimization criterion, and these three subjects, the required montage is extremely different. And if you look at that across, say, 50 subjects, again, you can see the color coding here means sort of how frequently is that electrode used, that we have very complex electrode montage patterns. And that's kind of quite impractical certainly in a clinical setting, but it's quite cumbersome. And another cost of this is that even though then we can control for the direction of current flow in the target area, the current there becomes really weak. So we're losing a lot of the intensity effect that we ideally like to get. Okay, so this was really just trying to highlight a few of the problems. We know that stimulation is variable, but what can we do about it? And to highlight just that there are a few variables we should probably control for, but that it's also not clear how to best do that. Personally, I think that for the whole debate about variability, that as long as we don't really control well enough what we deliver, the problem of variable outcomes is not going to go away. So I think it is a very likely a very substantial source of variance, I think. We don't really know what to control for. Should we focus on intensity, direction, focality, or all of it together? And whatever we optimize for, there's likely going to be a cost for that. And I think that while we could do this and figure that out, it's probably good to even step back and try to have a bit more and more compelling data that any type of current flow modeling actually makes a difference in terms of what the behavioral and physiological effects are. I don't think there's enough convincing evidence that if we do all these shenanigans, it really helps to control the variability. Of course, the problem that I think is always worth re-emphasizing and sometimes kind of falls by the wayside is that with any of this modeling, we can create great images, but it does not tell us anything about the physiological effect this has. So if we deliver the same intensity to a target area, we still don't know does that actually have what specific effect on the network activity that creates. As I mentioned at the beginning, I think most points really apply to all forms of non-invasive brain stimulation. And the two speakers this morning have, I think, greatly highlighted that as well. So I think these points I mainly list here to sort of perhaps initiate some discussions throughout the meeting and to point out that there's quite a lot of unanswered territory here, and certainly I think we're quite a bit away from knowing how we can control what we're delivering in an effective way. Thank you.